Alright guys, this is chapter 5, Carbohydrates. This is going to be part of the lecture and review. Once again, this is not a substitute for reading the chapter. You still have to read the chapter in order to understand what's going on. But before we do that, before we get into carbohydrates, I'm going to go ahead and talk about proteins and nucleic acids. Just a quick review. Remember, proteins have a diverse set of functions. For one, they speed up chemical reactions. They're a part of the immune system, and they're used by cells as a communication device, and they provide structure and function. Here are the different structures. For proteins, you have your primary structure, secondary structure, tertiary structure, and quaternary structure. Remember, all that starts off with amino acids, which is their monomer. Then we have our nucleic acids. Our nucleic acids down over here. You have DNA and RNA. Remember, DNA is double-stranded, has GCAT. RNA is single-stranded, which is a single-stranded copy of RNA, of DNA. Instead of having thymine, it has uracil. It's also made up of nucleotides. Nucleotides are made up of a phosphate, sugar, and base. And both of these guys hold their genetic instructions. This guy holds the genetic instructions is a copy of the genetic instructions. So that's just a quick review of the nucleic acids. Now we're going to get into our carbohydrates. All right, carbohydrates can be classified as simple or complex. So they're simple, they're going to be monosaccharides or disaccharides, as you can see the different ones here. And complex are the polysaccharides, which are starches, fibers, and glycogen, which we're going to get into in this review. The simple ones, glucose, fructose, and galactose, maltose, lactose, and sucrose. As you can see, they all end in O's. So that's a key thing to remember when either taking a test or doing the reading, is that anything that ends in O's will be a sugar. Okay. Now, monosaccharides, there's different ways you're going to see them. This is a long chain structure. It's usually made up of carbons. Here we have 6 carbons. That's 1, 2, 3, 4, 5, 6. 6 carbons, all right? Here we have a 6-ring structure. So we'll start off again. 1, 2, 3, 4, 5, and then 6 carbons, okay? So that's this ring structure of a monosaccharide, which is a monomer for carbohydrates, is a monosaccharide. Here's a long chain. It's still a monosaccharide, but it just doesn't have that ring structure. All right, here's a monosaccharide, a disaccharide, meaning 2, and a polysaccharide. And we're going to get into the formation of bonding, the polymerization, and so forth. All right, here are the monomers of monosaccharides, as you saw earlier. These are all simple carbohydrates. You have glucose, fructose, galactose, deoxyribose, and ribose. Glucose is our primary one. This is what's made by plants during photosynthesis. Fructose is it's a pentose sugar, because it has 5 sides, pentose. These guys over here are hexoses, or hexose, as you can see here, galactose and glucose. And then, of course, here we have deoxyribose and ribose. Deoxyribose, as you know, is found in DNA. And the difference between these two sugars, deoxyribose and ribose, is this location right here. This has a hydroxyl group. This just has a hydrogen. All right, that's the difference between deoxyribose and ribose. Once again, these guys are simple monomers, simple sugars. One thing you got to remember here is spatial arrangement. Spatial arrangement is the arrangement of the atoms around the structure. So for here we have glucose. Glucose is what we need for our primary source of energy. Galactose, on the other hand, it has a different arrangement of these molecules here. As you can see here, you have a hydroxyl group down at the bottom. Here you have it up on top. That small, minute change changes everything in how the molecule works. This is not a useful source of energy for us. So what we have to do, what our body has to do with galactose, is bombard it with some enzymes and change this to this in order for our bodies to use it. But before I get to that, I want to get back to this, the polysaccharides. Polysaccharides are long chains, or are long polymers, and I want you to remember there's disaccharides, and in between there's also called oligosaccharides. All right, oligosaccharides, which are a little bit smaller than polysaccharides, but not, and a little bit smaller than polysaccharides and larger than a disaccharide. Just wanted to cover that before I moved on. Once again, spatial arrangement and representation. Here we have different ways you'll see glucose. You'll see this structure sometimes, you'll see the ring structure, or you'll see the linear version. They all have 6 carbon sugars. Okay. Another thing is that is right here the beta and the alpha glucose. Okay, representation. This is the working form of glucose. Right here, when you have the OH in this position. Here we have it up on top, which is beta glucose. So once again, in order to use this, we'll use some enzymes to switch it over, and then it turns into the useful source of glucose, or a primary source of energy. So once again, the alpha glucose, as you can see here, the OH is up on top, and then when you go through that enzymatic activity, it can switch back and forth. It's a reversible reaction as well. You can also have this down here, the hydroxyl group down at the bottom. This is the functioning form of glucose. I apologize about the bell since I'm recording this during our lunch hour, but here we go. The polymerization of monosaccharides. Let me move this down over here. So here we go. Same thing as before, just as you've seen in the other biomolecules. You have your monomers. These monomers then form polymers through dehydration synthesis. All right, through dehydration synthesis.
Conquistadores, please disregard that bell. You guys still have about 15 more minutes. Please disregard the bell. Thank you.
Once again, guys, I apologize. I'll backtrack a little bit, the best that I can. Here's the polymerization of monosaccharides. You can't see, you can see it just a tad bit right there. Monosaccharides. So let me go back a little bit. Here we have a dehydration synthesis reaction, or a condensation reaction. You get the hydroxyl group of one monosaccharide and hydrogen from another monosaccharide. You remove it. That forms that bond. It's called the glycosidic bond. It forms a glycosidic bond between two monosaccharides. Having two saccharides is a disaccharide, and if you have anything more, is an oligosaccharide or a polysaccharide. So anytime you hear the word saccharide, that would be a key that it's a carbohydrate. All right. So once again, structure and formation determines the function and the function of the molecule. So here we have maltose and lactose. When you look at them all, they're 6 carbon sugar. They look like they have the same structure, but the bonds are different here. All right. Maltose is a sugar found in grains. All right. Lactose, of course, is a sugar found in milk. And this is the difference between them, is the way the bonds have formed between these glycosidic bonds. Here you have an alpha glycosidic bond in maltose and a beta glycosidic bond in lactose. And it would be 2 different enzymes to break these sugars down in order to form the glucose that we need. So if you're lactose intolerant, what the enzyme does, lactase is a protein, an enzyme that speeds up chemical reactions, and what it does, it breaks this bond right here so we can use the glucose. So what happens is, if you're lactose intolerant, you don't have the enzyme, or your enzyme doesn't function properly to break down the sugar, so the bacteria in your stomach does it, so the waste product of breaking this bond down is gas, so that's why you're a little bit gassy if you're lactose intolerant. All right, polysaccharide structure and function. Right here we have a little illustration that you see in your book, and this is a perfect one because it's going to let you know what each polysaccharide does and how it's different from all the others, and the way it's set up in function and in bonds. So starch is used for energy and is stored in plants such as potatoes. All right, you have an alpha 1,4 glycosidic bond, and they form these helixes. All right, and they can even branch out. So that's starch. Starch is found in potatoes. Then you have glycogen. Still, you have your alpha glucose. Your alpha glucose, remember, that's your primary source of energy. That's what our body needs for energy. Remember, it can't use the beta glucose, or it can't use galactose. It has to have alpha glucose. So it's the same thing as starch, but it's found in us. It's used in animal cells, and it's mostly found in the liver and in muscle. So glycogen is used during exercise. When you're exercising, your body starts using an enzyme called phosphorylase, and it breaks down this molecule to give you that glucose that you need for energy. And once again, they form these helix.
All right, guys, just bear with me. I put on a headset and make it easier for me to record. So I left off on glycogen. So starch is stored energy in plants. found in potatoes. Glycogen is found in us. It's stored in the liver and muscles. And then cellulose. Cellulose is completely different. This is a structural carbohydrate. It's used in the cell walls of plants and many algae. Algae are protistas, which can be unicellular and multicellular. Protistas can also be autotrophs or heterotrophs. They also have cell walls. Cellulose is a linkage of these beta glucose molecules. All right, so remember, our bodies need alpha glucose, or animals need alpha glucose for energy. Here now we have cellulose, which is a beta glucose. They have the same type of linkage, but now you start seeing some hydrogen bonding between the ring structures, between these carbohydrate chains. So they're parallel strands joined by hydrogen bonds, as you can see here. And once again, they make up the cell walls of plants and many algae. Now, chitin is also a structural carbohydrate. It supports cell walls of fungi or fungus, and it's also the external skeletons of insects and crustaceans. Insects and crustaceans, which I'll show you a couple of examples of. So basically the same thing, almost like cellulose. So you have these groups right here, but you have this one nitrogen group up here and down here as well. So when you see this right here, that's a little nitrogen group. It's a protein group, so it's made up of amino acids, so it's kind of like a protein. So that's where it gets its support from. And then there's hydrogen bonding, of course, within the parallel strands. All right. Then we have peptidoglycan. Peptidoglycan is found in bacterial cell walls. All right. And once we get into bacteria, we'll see how these cell walls are made, and kind of the same function as the chitin. You have these parallel chains, but then you see on here 4 amino acid forms peptide bonds, and some chains of amino acids are adjacent strands. So these are perpendicular instead of parallel, and are joined together by peptide bonds. So that makes their cell wall just a bit stronger than one made up of chitin. All right. So those are the structures of polysaccharides or carbohydrates. You have some that are used for energy, some for structure and support. So let's go over the ones for energy. So you know that glucose is made in plants. Plants are autotrophs. They're photosynthetic. And I'll give you a rundown of what happens in the photosynthetic process. You have light, water, and carbon dioxide. The plant takes all that into the chloroplast, right? In the chloroplast, you have two types of reactions going on. You have light independent reactions, which are the dark reactions, and the light dependent reactions, which are these over here. And the light reactions produce oxygen, but then the light independent or dark reactions produce sugars. These sugars are used in cellular respiration to make cellulose, starch, and other organic compounds. So photosynthesis is key to life on this earth. Okay, that's where most of our energy comes from, or energy comes from. And when you make these sugars, just remember that all the energy from the sun is stored within these sugars. Okay? And then those sugars, this is for us, we have those eukaryotes that have mitochondria. Those sugars are then used in the cellular respiration process to produce. Once again, I apologize for this, but I'm going to keep on going because I've already invested too much time into this lecture review. Once again, you get glucose. Glucose has all the energy from the sun stored in its bonds. Glucose is then broken down by the mitochondria in order to produce ATP, which is our energy coin. All right, and that's done in the mitochondria, and we'll get more into that once we we'll move on into in the year. But just know that all energy comes from the sun, and it drives these reactions inside the chloroplast, which produces sugars. These sugars are then used by us to produce ATP, and our mitochondria does that via cellular respiration. Here we have stored energy, as we spoke before. Here we have starch. Here are the rings of starch, and they start forming those helix or helices. Here we have the liver, which stores glycogen. All right, so stored sugars in plants is starch. Stored sugars in us is glycogen. For structural and support, this is chitin. Here's the exoskeleton. You know how some insects molt. That means they lose their outer skeleton once they grow out of it. So it's made up of chitin. Here's some fungi. Their cell walls are made up of chitin. And every time I think of cellulose, I always think of celery. Celery, that fibrous material inside the celery, is made up of cellulose. Okay, so those are 3 structural carbohydrates. all in one. And also polysaccharides, they're part of their structure and function. Let's wait for the bell here. Part of their structure and function here is, I should have changed this to signaling and cell recognition. So what's happening here, this is your cell membrane. So each one of our cells has a cell membrane. That's what makes a cell a cell. And the cell membrane is made up of 2 layers, a phospholipid bilayer. But within that bilayer, you're going to find all these different pieces that are essential to the cell's existence or the cell's life. So for example, here we have a glycoprotein. This is a protein, and this is anything with glyco in it, like glycogen, has to do with carbohydrate. So here you have a carbohydrate chain. They're like the little antennas of the cell. They're used for signaling cell-to-cell interaction, or also what's happening in the environment. They send messages to inside of the cell. Once again, this is a glycolipid, so instead of being attached to a protein, it's attached to a lipid, because remember, like I just mentioned a few seconds ago, your cell membranes are made up of phospholipids, so it attaches to a lipid here, and it's a carbohydrate chain, so it's a glycolipid. Once again, these guys are like, I like to call them the little antennas. They allow communication with the outside environment of the cell, or either communication between cells. Move my thing over here so give you a better view of it. Also, cell identity and signaling. Your blood type is also based on these little sugar residues outside the red blood cells. So depending on the blood type you are, you're blood type O, B, or A, you're going to have a different set of sugars on the outside of your red blood cells. And that's it for now, guys. That's it for carbohydrates. I apologize for all the interruptions, but like I said, I've invested too much time into it, and I was doing it during our lunch hour, so I hope you enjoy the rest of your weekend. Remember, this is not a substitute to reading the book. You still have to read the book. This is just to give you a different view and a different perspective of what's in the book, and hope you understand the content and the material in the book. So I'll be sending you guys your reading questions. Probably no vocabulary, since most of the vocabulary I covered it during the lecture, but be waiting for that assignment to come in, and if there's any suggestions on what I can do a little bit better, let me know and I'll fix them. Thank you, guys.